Welcome back to IGCSE Science Academy. Hello everyone. Today we are going to continue the topic Enzymes Part 2. In our previous video we covered topic 5.1.1 and 5.1.2. Today we are continuing the unit Enzymes by completing 5.1.3 Importance of enzymes, 5.1.4 Enzyme action, 5.1.5 Temperature and pH. Don't forget to check the previous video. The link is given below to help you stay on track. Let's get started and make science simple and interesting. Dear students, think of your body like a massive factory. Every second, thousands of chemical reactions need to happen. But without help, they had to be too slow to keep you alive. That's where enzymes come in. Today we are defining exactly what they are and the three things that make them so special. Let's define the enzymes. Enzymes are biological catalysts, proteins, molecules that speed up chemical reactions in living organisms without being used up or permanently changed. The three key features. First is made of protein. Enzymes are globular protein with a specific 3D shape formed by chains of amino acids. Second, highly specific. Each enzyme only catalyzed one specific reaction due to its unique active site shape. Third is reusable. Enzymes are not consumed during the reaction. They can be used again and again. Once an enzyme finishes one job, it releases the product and moves straight to the next substrate. You don't need millions of enzymes, you just need a few that work really, really fast. So topic 5.1.3 from unit enzymes. Why are enzymes important? We know what enzymes are, but why do we actually care? Why are enzymes important? Reactions must occur fast enough to sustain life. If our body if our body's chemistry ran at its natural speed, we wouldn't survive the next 5 minutes. Here is why. First, let's look at body temperature. 837 degrees centigrade uncatalyzed reactions are far too slow for metabolism. Enzymes increases rates by millions of times. So enzymes are the turbochargers here. Next is this is vital for our metabolism. All metabolic processes, respiration, digestion, photosynthesis, protein synthesis require enzymes. Every single process from respiration, releasing energy and digestion, breaking down food, to photosynthesis in plants and protein synthesis in our muscles requires enzymes. Without them, metabolism just stops. Now think about coordination. Thousands of different reactions occur simultaneously in cells, each controlled by specific enzymes. Inside just one of our cells, thousands of different reactions are happening at the same time. How does the cell keeps them organized? Of course, because enzymes are specific, each reaction is controlled by its own enzyme. This allows the cell to coordinate complex processes without overthinking, turning into chemicals. Finally, there is energy efficiency. Enzymes lower the activation energy needed to start reactions, saving the cell's energy resources. To get a chemical reaction started, we usually need a spark or a push. This is called activation energy. Enzymes lower this energy barrier by making it cheaper for the reaction to start. They save the cells precious energy resources, they allow life to happen at a lower energy cost. So enzymes are not just protein molecules, they are the engines of life. Dear students, now listen carefully to understand the activation energy. If you want to start a fire, you need a spark. In biology, if you want a chemical reaction to start, you need a spark of energy that is called activation energy. But there's a problem. Your body doesn't have enough heat to provide that spark for most reactions. That's where enzymes perform their magic. Let's look at the graph. First, let's define its activation energy. That is EA. EA represents activation energy, the minimum energy required to start a chemical reaction. Think of it like a giant hill that molecules have to climb over before they can turn into products. If they cannot get over the hill, the reaction never happens. Then life without enzyme. High activation energy barrier, very slow reaction rate, insufficient for life. Look at this red line. This is a reaction without an enzyme. See how high that peak is. That's a high activation energy, that is EA. Barrier age normal body temperature 37 degrees centigrade, molecules simply don't have enough kinetic energy to get over that massive hill. The result, the reaction is way too slow to keep you alive. With enzymes, enzyme lower activation energy, reaction proceeds rapidly at body temperature 37 degree centigrades. These green solid lines on your graph, now look what happens when we add an enzyme. The enzyme lower the activation energy. It effectively shaves off the top of the hill because the hill is much lower. Many more molecules have enough energy to get over it, even at 37 degrees centigrades. The result, millions of reactions per second become possible, sustaining cellular life processes. Millions of reactions that were previously impossible at body temperature now happens in a fraction of a second. This allows your cell to sustain life, repair damage, and release energy instantly. Without lowering that hill, life as we know it would stop. Remember, enzymes is equal to activation energy, EA is equal to faster reaction. Topic 5.1.4 from unit enzymes, that is enzyme action, the lock and key model. We have already discussed in detail with diagram in our previous video. You can watch from there also. If you have ever tried to open your front door with your car keys, you know they don't fit. Enzymes work the exact same way. This is the lock and key hypothesis, and it's the reason why enzymes are so specific about which reactions they help. Before we look at the steps, let's meet our players. The active site, substrate, enzyme substrate complex, and product. Active site specific region of the enzyme where the substrate binds has a complementary shape to the substrate. This is the log. It's a specific region on the enzyme with a very precise 3D shape. Substrate, the reactant molecule that fits into the active site of a specific enzyme. This is the key or the reactant that needs to be changed. Then enzyme or substrate complex. Temporary complex formed when substrate binds to the active site. This is the most important phrase for the enzymes. The substrate isn't the same shape as the active site, it's complementary, meaning they fit together perfectly. And the product. The molecules releases after the reaction the substrate has been converted. Finally, the substrate is converted into products. They are released from the active site because they no longer fit the shape. Notice the enzyme, it's completely unchanged and ready to go back to step 1 to do it all over again. So here are the 4 steps. Lock and key model. First is free enzymes, active site empty and available. Second is substrate approaches, complementary shape fits in. Third is enzyme substrate complex, bond forms, reaction occurs. Fourth is product released, enzymes free to act again. And that is the lock and key model in 4 easy steps. Just remember, the enzyme is the lock, the substrate is the key, and they must be complementary. Dear students, hold on and think about this statement. Why does not a digestive enzyme meant for protein starts breaking down your DNA? The answer is specificity. Just like you cannot use a house key to start a car, an enzyme is locked to only one type of substrate. Let's discuss about specificity, complementary active site shape. The most important thing to remember that is the active site shape is complementary, not identical to the substrate like a lock and its key. Note the word complementary, not identical. They fit together like two matching puzzle pieces. But where does the shape come from? Remember, enzymes are protein. Their specific 3D shape is determined by their amino acid sequence. Only a substrate with a complementary shape can bind to form an enzyme-substrate complex. A different substrate wrong shape cannot bind. It's explain enzyme specificity. The enzyme is not changed by the reaction. Its active site remains available for more substrate molecules. Let's look at the diagrams. On the left, we have the correct substrate. It's a perfect match for the active site. It fits. An enzyme substrate complex forms and release, and the reaction proceed. On the right, we have a different substrate, the oval. Even though it's nearby, it has the wrong shape, it cannot bind. This is exactly why enzymes are specific. They literally ignore molecules that don't fit their active site. And just like the lock and your door doesn't change after you use your key, the enzyme is not changed by the reaction. Once the product leaves that specific active site is immediately available for the next matching substrate to arrive. Topic 5.1.1 from Unit Enzymes Effect of Temperature on Enzyme Activity. Why do we get a fever when we are sick? And why can a very high fever be factor? It all comes down to how temperature controls your enzymes. Let's look at the 3 stages of enzyme activity as the heat turns up. Low temperature 0 degree centigrade to below optimum. Low kinetic energy in molecules, fewer effective collisions between enzymes and substrate, slow reaction rate but enzyme not denatured, warming up restores activity. At low temperatures, specifically below the optimum, everything is moving in slow motion. Molecules have low kinetic energy, so they are not moving around much. This leads to fewer effective collisions between the enzyme and the substrate, and the result a very slow reaction rate. Notice that enzyme is not denatured here. If you warm it up, it starts working perfectly again. Second is optimum temperature, that is 37 degrees centigrades in human. Maximum kinetic energy for efficient collisions, highest rate of effective enzyme substrate collisions, maximum enzyme activity, active site shape is ideal. We hit the optimum temperature in humans. This is usually around 37 degrees centigrades. Here, molecules have the maximum kinetic energy for efficient, frequent collisions. We have the highest rate of effective enzyme substrate collisions. At this point, the enzyme activity is at its absolute maximum and the active site shape is ideal for the substrate. Then high temperature above optimum. Excessive kinetic energy disrupts bound. Active site changes shape, denaturation. Substrate can no longer fit, irreversible enzyme permanently destroyed. What happens when we go above the optimum? This is the danger zone. The excessive kinetic energy becomes to disrupt the bonds that hold the enzyme's 3D shape together. The active site changes shape and we call this denaturation. Because the shape has changed, the substrate can no longer fit. Denaturation is irreversible. Unlike the cold stage, cooling down a denatured enzyme won't fix it, it's permanently destroyed. Effect of pH on enzyme activity. Imagine taking a fish from the ocean and putting it into a pool of vinegar. Of course, it wouldn't survive, right? Enzymes are the same way. They are incredibly sensitive to how acidic or alkaline their environment is. Let's look at why pH can make or break an enzyme. Every enzyme has the optimum pH where it works at its absolute best in humans. Most enzymes like to stay near pH 7 or 8, but there are some famous expectations. Now let's look what is pH. pH measures the concentration of hydrogen ions, scale 0 to 14, acidic 0 to 6, neutral 7 and alkaline 8 to 14. What is the optimum pH? Each enzyme works best at a specific pH. Human enzymes typically work best near pH 7 to 8. pH too high or low. Hydrogen ions interfere with hydrogen ionic bonds holding the enzyme's shape. Active site distorts and denaturation. What happens if we move an enzyme away from its happy place? Those H-positive ions we mentioned earlier start to interfere with the hydrogen and ionic bonds that hold the enzyme's 3D shape together. Just like with high heat, the active site distorts and changes shape. This denaturation, once the active site is distorted, the substrate can no longer bind and the reaction stops. Look at these examples of enzyme optimum pH. First is pepsin found in our stomach. It's actually work best at very acidic, that is pH 2. Then second is salivary amylase. In our mouth it stays right at neutral pH 7. Trypsin working in our small intestine, it prefers a slightly alkaline, that is pH 8. And then catalase, that is present in our liver cells, and the pH is neutral for catalase, that is 7. Every enzyme has an optimum pH where it works at its absolute best. In humans, as we have discussed earlier, most enzymes like to stay near pH 7 or 8, but there are some famous expectations. Now let's understand the concept of denaturation. Denaturation, an irreversible change to the shape of an enzyme's active site caused by high temperature or extreme pH, preventing the substrate from binding. So the cause for the denaturation is high temperature or extreme pH level. The effect is the active site changes shape, preventing the substrate from binding. So now let's differentiate the normal enzyme and denatured enzyme. Normal enzyme active site intact, 3D shape maintained by hydrogen bonds, active site has correct complementary shape, substrate can bind successfully, reaction rate at maximum. Denatured enzyme active site changed, excessive heat, extreme pH breaks bond, 3D shape of protein irreversibly changes, active site shape is distorted, substrate can no longer bind, no catalysis. Let's discuss about homeostasis, why the body maintains constant conditions. Think about it, why do we sweat when it's hot and why do we shiver when it's cold? It's not just to make us comfortable, it's a life or death. It's a life or death mission to protect our enzymes. This is the world of homeostasis. The human body maintains temperature at 37 degrees centigrade and blood pH at 7.35 to 7.45 to keep enzymes at their optimum conditions, essential for metabolism and survival. If these levels drift too far, our metabolism fails and survival is at risk. Temperature regulation. Your brain has a built-in thermostat called the hypothalamus. Hypothalamus monitors and regulates body temperature. Sweating cools the body when too hot. Shivering generates heat when too cold. 37 degrees centigrade is optimum for most human enzymes. Fewer 40 degrees centigrade can dangerously denature enzymes. If you get a fever above 40 degrees centigrade, your enzymes are in the danger zone where they can begin to denature. pH regulation. pH is just as important. Your body uses several tricks to keep pH stable. Lungs regulate carbon dioxide levels, affects blood pH. Kidney excrete hydrogen ions to control blood pH. Blood buffers, for example, carbonates, bicarbonates, resist pH changes. Stomach maintain pH 2 for pepsin to work. Small intestine has alkaline bile to neutralize acid. Notice that while blood is neutral, the body creates specific extreme zones like the stomach maintaining pH 2 for pepsin to work or the small intestine using alkaline bile to neutralize that acid so other enzymes do not denature. Now let's summarize the lesson and discuss the key points and vocabulary. Enzymes, biological catalysts made of proteins, speeds up reaction without being used up. Substrate, the reactant molecules that binds to the enzyme's active site. Kinetic energy, energy of motion in particles. Higher temperature is equal to more kinetic energy is equal to more collisions. The temperature or pH at which an enzyme works at its maximum rate. Active site, the specific region of the enzyme where the substrate binds, has a complementary shape. Enzyme-substrate complex formed during the reaction. Denaturation, irreversible change to active site shape caused by high temperature or extreme pH. Homeostasis, maintaining a constant internal environment essential for enzyme function in the body. At last, let's discuss about the exam tips and the common mistakes and try to avoid those common mistakes. First is active site language. Always say the active site is complementary to the substrate, not the same shape or identical. These are different things. Second is denaturation as inhibition. Denaturation is irreversible, permanent shape change. Inhabitation is reversible. Don't confuse them in exam answer. Third is temperature and kinetic energy. Link temperature to kinetic energy then to frequency of effective collisions. Use all three terms in your explanation. Fourth is pH and denaturation. Extreme pH changes the shape of the active site by disrupting ionic and hydrogen bonds leading to denaturation. 5 is optimum as maximum. The optimum temperature pH gives the maximum rate of reaction. Learn to describe and sketch the bell-shaped actively curve. 6 is enzyme not changed. Emphasize that enzymes are not used up. They are catalysts and can catalyze many reactions one after another. Thank you for watching. For the complete topic-wise explanation of unit enzymes, don't forget to subscribe.